I want to point out really how complex tumors are as we think going forward about therapeutic options. So we think of tumor cells as tumor cells, all the same, working the same way with many of the same genetic changes. But the reality is, tumors actually evolve through the course of time. And so it becomes more of an ecological composition of subpopulations of tumor cells that have developed new mutations and new ways of competing and winning in the niche that is the tumor. So a tumor cell is not a tumor cell across the board. And so when we think about what a tumor biopsy is, we think of it being a piece of tumor tissue that is almost all tumor tissue that we can understand with sequencing and other techniques. But I want to point out to you how really complex this whole system is. I'm going to overlay now just the lymphocyte components of the tumor microenvironment. And these include cells that are possibly trying to kill the tumor in CD8 positive cytotoxic T cells, cells that are helping to direct those in CD4 positive cells, B cells that might be reacting to tumor antigens and producing antibodies against the tumor, which can be potentially anti-tumor or can actually block potential therapeutics of the tumor. Natural killer cells that are present that recognize these tumor cells as abnormal because maybe they don't express enough MHC1, which is supposed to be the general identification card of the protein contents of most cells. And if cells are hiding that information, the immune system through natural killer cells becomes suspicious and will eliminate them. And also clear bad guys like T regulatory cells that are blocking the immune attack against the tumor and are contributing to the ability of that tumor to survive in the face of an anti-tumor immune response. And it's even more complex than that because there are T helper 17 cells and new subpopulations of lymphocytes are being discovered all the time. Now, on top of that, we'll add the monocyte macrophage myeloid lines of cells. And these include macrophages that are polarized classically into either M1 pro-inflammatory, potentially anti-tumor macrophages, or M2 macrophages that are more immune suppressive, immune reductive, causing the immune system to try to settle down and reduce inflammation in the area, thereby protecting the tumor from that immune response. Also, myeloid-derived suppressor cells are present in the area that migrate into the region, are very potently suppressive of the immune system in that region. And all of these interact substantially with one another to where the tumor cells have caused the immune system to develop a protective shield around it to let it expand in the face of an anti-tumor immune response against the mutant proteins that these tumors inevitably carry. And it finally gets even more complex than that when we add normal stromal cells, just fibroblasts and other cells in the environment that can not only support the tumor cells physically, but also even produce important products that help the tumor cells grow new blood vessels, help the tumor cells reduce the immune response, and help the tumor cells invade the local stromal tissue in order to expand and gain new territory. And so you can see if you were to take a biopsy of this tumor, only a portion of the cells in that biopsy piece are going to be tumor cells. And in fact, pancreatic cancer in humans is probably the champion of lowest concentration of tumor cells. As few as 17% of all the cells in a biopsy of pancreatic tumor are actually tumor cells. The rest are these other stromal cells and immune cells that are present in the microenvironment. And so all of these together interact to create an environment that protects the tumor, but also offer us opportunities to intervene and try to reawaken the immune response and kill the tumor. Thinking of these constituents, then, I want to point out 4 different immune strategies that are frequently used.